Okay, so let's look at question 19. That's the next one that you asked for questions about. So here, I wanted you to find information about this gene. And you're like, I don't know how to find information about this gene. This is the last bioinformatics tool we did. So actually, let me share the screen a different way so that you can see. I'm going to have to open more tabs. So here we go. Okay, so we are here, and I want to find information about that gene. And okay, that's the one acronym you need to remember. That one, or write it down somewhere, is NCBI. So we want to find information about that gene. They're like, here, click gene. And search that gene. No, I guess I didn't copy it. I want to copy that. And did I say I want the human one? Mutations have been neurological. Okay, yes, I did say human. All right. So come on, show the suggestion again. All right, that's all right. So here the first one is often the first one will be human because that's what people are studying or more interested in. So you click on that. It's going to take you to this screen which shows you where, in which chromosome it is and which arm of the chromosome, where more or less in the chromosome. And then here in red, you'll see the gene you're studying. And this gene is weird in terms of normally for eukaryotes, you expect a gene and then another gene or a bunch of space in between them. But you don't expect a gene within a gene, which is why I thought, oh, this is so interesting. And you're like, how can the same DNA fragment encode these two genes that SLC, whatever, and see that? That's like the arrow there. And it's because this gene is encoded in one DNA strand. We're going to call it the Watson strand because it's going from left to right, which means it's, if you go from lower nucleotide here to a higher, so a 5' to 3', it's encoded in that strand, left to right. So that's the coding strand for that gene. This other one, right to left, it's encoded on the other DNA strand. So the mRNA sequence is the reverse complement of the other DNA strand, not this one. So they are on the same location, part of them are the same location of the genome, but one in one strand and one in the other. Okay, so let's see what the question is asking. Let's see, where are the SNPs most concentrated? So what I wanted to do is to go down here and find the SNPs. And I asked you to, so in class we did the NCBI Homo sapiens annotation. So this one is the gene that we are looking at. And then this is something else. See how it's pointing left? If you hover over, you'll see it's this other gene here. So this gene was like confusing with all these arrows. That's why it's in homework, not in exam. So the gene we're looking at is this one with the boxes. And then the skinny lines are the introns. And then you'll see this gene doesn't appear to have introns. So if I click on it, it will expand and it will say, this part here is the coding DNA sequence. So if you translate that, that's your protein sequence. This part here has more, so that's the mRNA. This middle part is the part of the mRNA that will end up in the protein. So that's the coding. This middle part here is the coding. Then above it is the 5' UTR. Below it is the 3' UTR. And that's what the light green means. It's the 5' and 3' UTR. And then I asked you, where are most of the SNPs? So there are many SNPs in here. That's what you can see. And then the clinical ones are these ones. Not all of these are of clinical relevance. So I ask you, where are the SNPs? And then let's see if you can put on the chat, just because it's faster. So mostly what the goal of this question is so that you can see what is the gene and what are the parts of the gene. If this part is the coding, then the darker green is the exon, that's the coding sequence. The light colored part is the 5' UTR, this is the 3' UTR. And the SNPs, you look at these tracks here. This is a bunch of SNPs, and these are the clinical SNPs. So they are in the coding DNA sequence. I think you also asked for question 20, and since I'm already on this screen, I said what type of mutation is that let's see, is found when you examine the track. Okay, you actually need to add this track because it will show you the clinical ones, so the ones that are pathological or not. So to add a track, you go here on tracks, then you go, so tracks, configure tracks, and you're going to search tracks, and I'm just going to paste this here. So in an exam, if I ask you about that specific track, I'll give you that name. You don't have to remember it. Okay, so that's the track I want. You click on that, and you click here, configure down here, so that it adds that track. So this one is the track that got added. Come on, close that. So this one is the track that we just added, and I'm asking specifically about this one SNP. So if you hover over, this screen shows up, and you click on the variation here, and you will see that it's not a variation that affects the DRD5 gene. It's actually looks like it's affecting the other gene. So you click on this. Let me see if it's opening the screen you need to see. And then I'm asking you, what type of variation is that? So I wanted you to see, okay, for this gene, it's saying it's there. So in here it says it's intron variant, but I want to see not on that gene, I want to see on the DRD5. So most of them are talking about the SLC, whatever. I mean, like, oh, here on the DRD5, so there is an effect on the other DNA strand too, and the change goes from a serine to a phenylalanine because there's like one nucleotide difference, and when there's one nucleotide difference, this one SNP caused a change in amino acid, so it's a missense variant. And that's the answer for that question. That's a missense.